Hello friends, I'm Dr. D.P. Bora. I welcome you all to my YouTube channel, The Microbiology Digest. Today we will be discussing another new chapter in microbiology, that is the organisms under the order Rickettsials or Ricketsias. Now, the order, as I said, it is Ricketsialis. So this Ricketsialis orders order. They are also informally they are called as Rickettsias. They are a group of small alpha proteobacteria. Now they are obligate intercellular parasites, and some are very notable pathogens. For example, Rickettsia, that is a pathogen for human and animal, and another one is Ehrlichia, which is mostly responsible for causing diseases in animals. Now, when this group of organisms were discovered, so it was very confusing. The properties or characters of these organisms, it it was found to be very confusing to which group they should be included, whether they are bacteria or they are viruses, because some of the properties of this Rickettsia group of organisms, for example, they are minute intracellular parasites, obligate intracellular parasites. So, which is an essential character of viruses. Then another one, they require a living system for multiplication. This is also same character like viruses. So that is why they were formerly considered as closely related to the virus. But again later on, few other characters which were found to be uh, having resemblance to the bacteria. For example, their cell wall structure is similar to gram-negative bacteria. They divide by binary fission, just like bacteria. They contain cell wall, which is composed of muramic acid. Then they possess both DNA and RNA. Both types of nucleic acids are present. They are visible under light microscope. So these are, and they are susceptible to some of the antibiotics. So these criteria, uh, when these properties were studied, so it was found that they are not related to virus, more closely related to the bacteria. So they were placed as a bacteria under bacteria, but they are a special type of bacteria which need a they are called as obligate intracellular parasites. Okay, now how these organisms were discovered? So this the name Rickettsials, it was given to this group of organisms in the honor of American pathologist Howard Taylor Ricketts, who first observed this type of microorganisms in Rocky Mountain spotted fever. This is a disease caused by Rickettsia, one organism under Rickettsia group. So Taylor Ricketts, he was the first person to identify this group of organism in the cases of Rocky Mountain spotted fever. And this was in the year 1909. Then later on, in the year 1935, another scientist, Derrick, so he investigated the cases of fever which was occurring in abattoir workers in Australia, in Brisbane, Australia. So a specific type of disease or fever was going on during that time, and this fever was confined to the abattoir workers only. So it was studied by Derrick in the year 1935. And when he studied this disease or fever in abattoir workers, the etiological agent responsible for this disease was unknown. Okay, so that is why, as the etiology was not known till that time, so the disease was named as quarry fever. Okay, with unknown etiology, it was said as quarry fever. And from that quarry fever, the word Q fever, which is a disease caused by Rickettsia, one of the Rickettsia organism. So the disease name was given as Q fever later on. And the causative agent of this disease, quarry fever or Q fever, it was identified by two scientists. One is Barnard and Cox. So, in the honor of these two scientists, as they were successful in identifying the positive agent, the organism was named in their honor as Coxiella barnetti. Now, what are the general properties of this group of organisms? So, Rickettsia, they are small, non-motile, non-capsulated, pleomorphic, and coccobacillary organisms. And they are intracellular, obligate intracellular parasite. They exist as obligate intracellular parasite. Then, under electron microscope, Rickettsia are seen to have three-layered cell wall. They have a three-layered cell wall and trilaminar plasma membrane. So this resembles the structurally they resembles the gram-negative bacteria. So already we have studied the structure of gram-negative bacteria. So structure of the Rickettsia organisms, it resembles more closely to the gram-negative organism. Then they stain well with common stains Romanowsky methods like Zimsa stain, Castaneda, then Lissman stain, Machiavello. These are these organisms, they can be stained very well, but the stain, if we go for gram staining, they stain very poorly with gram stain. Now coming to the habitat. So they are essentially, Rickettsias are essentially parasites of arthropods. So they replicate mostly in the cells of the gut. And these organisms, they can be passed to the next susceptible host, either transovarily or transstadially. Both type of transmission can be possible in case of Rickettsia organisms, and they do not survive outside the living cells, as they are obligate intracellular parasites. They cannot survive outside the living cell, but there is one exception for one organism, that is Coxiella barnetti. It can survive by forming some endospore like form. This is the only Rickettsia which can survive outside the host living cell. Why they can survive? Because they can form a endospore like form, and thus they can remain viable outside the living cell. So this is only possible for Coxiella barnetti, the causative agent of Q fever. So, and some of the Rickettsias, it has been shown that they can exist in the host in the latent form. So, latent form means they can remain in the host without exhibiting any signs and symptoms of the disease. Now, coming to the classification. So, as we have discussed, these are usually parasites. Rickettsias are parasites of alimentary tract of most of the arthropods. For example, fleas, lice, ticks, and mice. Mites. So this group of organisms, they can be transmitted from arthropod only to the animal or to human. So they are classified based on the properties like the disease they produce, the vectors by through which they can be transmitted, and antigenic relationship. So these properties are taken into consideration while classifying the Rickettsia group of organisms. So family order Rickettsials, they are classified into two different families. One is Ricketsiaceae and another one is Anaplasmataceae. So again, Ricketsiaceae family, it has been divided into classified into five different genera. So first genera is the Caudria, then Ricketsia, Coxiella, Arlichia, and Neoricketsia. These five are the genera under the family Ricketsiaceae. Then second family, Anaplasmataceae, they are again uh, classified into four different genera. First one is Anaplasma, then Aegyptianella, Apirithrozoon, and Hemobartonella. So these four are the genera under the family Anaplasmataceae. And these organisms, each and every organism under Ricketsiaceae as well as the Anaplasmataceae family, they parasitize on a specific cell type in the host. For example, Caudria, they are mostly targeting the vascular endothelium as well as the macrophages. Then Rickettsia, macrophages, leukocytes, and endothelial cells. Coxiella, mostly the circulating lymphocytes. Then Ehrlichia, this is vascular endothelial cells again. Then Neoricketsia, these are reticular cells of lymphoid tissue. These are the cells where the organism prefers to multiply. Then similarly for Anaplasma, almost all Anaplasma, all the organisms under Anaplasmataceae family, they mostly replicates or parasitize in the erythrocytes. Now coming to the cultivation of this group of organisms. So as we have already discussed, Rickettsia, they can multiply by the binary fission method. So they employs the binary fission method like bacteria for multiplication. So they have the cytochromes and their metabolic reactions are aerobic. So these properties mostly related to the bacteria. Then they possess many of the metabolic functions of bacteria as we have already discussed, and they require some exogenous cofactors in the from the animal cells. And further, these Rickettsia organisms, as we have already discussed, they are obligate intracellular parasites. They require living cells for replication. So unless and until living cells are provided or they come in contact with the living cells, this group of organisms they cannot multiply. So they can be cultivated in the laboratory by using the yolk sac of the developing chicken embryo, and we can use some of the cell lines, established cell lines like HeLa cell, HEP2 cell, and mouse fibroblasts. These are the cells which are commonly used for cultivation of Rickettsia organisms in the laboratory. And the growth of the organisms, Rickettsia organisms, they mostly occur in the cytoplasm of the infected cell, and also in some cases the multiplication may occur in the nucleus. For example, in case of spotted fever group, the organism multiplies in the nucleus of the infected cell. Then Ehrlichia canis, another organism under Rickettsia group, they can be propagated very well by using dog monocyte culture. So dog monocytes, if we culture in the laboratory, so we can use monocytes from the dog. They can be used for cultivation of the Ehrlichia canis. Then here is one exception. So there is one organism, that is Rosalimia quintana. This is the only Rickettsia which can be cultivated in the artificial media. Means this organism, this is an very uh, good exception from all the Rickettsia organisms. Means Rosalime quintana is the only Rickettsia which have the ability to grow in blood agar. So this is to be noted very well, because this is the only Rickettsia which can grow in blood agar medium. Then for if we talk about the animal models for primary isolation, mostly guinea pigs and mice are used for as an animal model for cultivation of the uh, or primary isolation of the Rickettsia organisms. Now coming to the antigens and toxins of these organisms. So basically three types of antigens have been demonstrated: group specific soluble antigen, then species specific antigen, and third one is the alkaline stable polysaccharide. So this is a very important antigen which is also demonstrated, or which is also present in some of the bacterial strains of Proteus. And this uh, antigen is the basis of the well-Felix reaction, which is a diagnostic test for rickettsial diseases. So three strains of Proteus has been found to contain this polysaccharide antigen, that is Proteus OX19, OX2, and OXK. These three strains are found to uh, have this uh, polysaccharide antigen. Now coming to the diseases caused by this group of organisms. So these rickettsial organisms, they are associated with specific diseases in both men and animals. So for example, Cowdria ruminantum. This organism, Cowdria ruminantum. This is associated with a specific disease called heart water, and main host ruminants are mostly affected, and transmission is through ticks. Then Rickettsia prowazekii. This is associated with this is the causative agent of epidemic typhus, a disease in human, and this is transmitted by louse. Then Rickettsia typhi. This is again associated with epidemic typhus. The next one is Rickettsia museri. This is associated with another disease condition called endemic typhus. This is also in human, and this is transmitted by rat flea. Then Rickettsia rickettsii, it is the causative agent of Rocky Mountain spotted fever. This is in human, dogs, as well as in rabbit. And ticks, mostly the dermacentor species of ticks, the uh, they act as a vector for transmission of this disease, organism. Then next, Rickettsia susugamosi. So this is associated with scrub typhus. Mostly the small rodents and birds, they are the main host of this disease and transmitted through mites. Then Rosalime quintana. This is the causative agent for trans fever, mostly in human, and this is transmitted through louse. Then last Rickettsia organism is the Coxiella barnetti. This is not last, sorry, Coxiella barnetti. It is associated with our causative agent of the Q fever, which is a zoonotic disease having very high zoonotic importance and transmitted by ticks. Next organism is the Arlesia group of organisms, Arlesia bovis, Arlesia canis. They are also transmitted through ticks, and Arlesia bovis is associated with bovine ehrlichiosis. Then Arlesia canis is associated with canine ehrlichiosis. Then this is also otherwise known as tropical canine pancytopenia. This is the other name of canine ehrlichiosis. Then Ehrlichia phagocytophilia. This is associated with a specific disease condition called grazing fever in cattle. So Grazing fever in cattle is caused by Rickettsia phagocytophilia, and this is also transmitted through ticks. Then Rickettsia rstc, it is associated with Potomac horse fever in horse. Then last one, the Rickettsia helminthica, it is associated with a specific disease in dog. It is called as salmon poisoning. So salmon poisoning, it is the dogs mostly they get infected due to the ingestion of salmon fish containing the infected helminth flu, where these Rickettsia organisms they present, and the disease in dogs is caused by it is called as salmon poisoning in dogs caused by Rickettsia helminthica. Now coming to the pathogenesis of these diseases. So basically in the pathogenesis there are six specific steps. Number one is the adhesion. Then second one is the internalization of the organism. Then internalization is through formation of a phagosome, and the organism, once it is internalized, the organism will be released to the cytoplasm from the phagosome. Then there will be intercellular growth of the organism. Then once the organism gets fully matured, they will be released from the infected cell so that they can infect a new cell, susceptible new cell. Then last one, the host will respond. Host immune system will definitely uh, try to respond against that infection. These are the main basic steps in the pathogenesis of rickettsial diseases. Next, coming to the diagnosis of these diseases. So samples will have to collect the unclotted blood, affected tissues, paired serum samples, and in case of Q fever, sputum and urine also uh, we can detect the presence of the causative organism. Then we can go for direct microscopy, and we can go for isolation and cultivation, although it is very difficult and not generally uh, practiced, but still we can go for the isolation or primary isolation by using animal models. Then if it is the disease is the organism can be recovered from the infected animals, so we can confirm the disease. Then another very important test that is called as Well-Felix reaction. So we have discussed earlier preliminarily. So Well-Felix reaction, it is one of the very important serological tests. It is done for diagnosis of most of the rickettsial diseases, and this is actually an agglutination test. And this test was developed by two scientists, Weil and Felix, in the year 1916, and in their honor the test has been named as Well-Felix reaction. So what happens, as we have already discussed, protease group of organisms, they share some polysaccharide antigens which is also present in the Rickettsial organisms. So Proteus vulgaris and Proteus mirabilis, these two organisms, Proteus vulgaris OX19 strain and OX2 strain, and from Proteus mirabilis OXK strains. So they share some of the polysaccharide antigens, and which can be used for agglutination test between the rickettsial organism as well as the proteus. So the in case of specific diseases, the specific strain of the bacteria proteus organism, they give the positive reaction. For example, if it is epidemic typhus, then OX19 will give the agglutination. Endemic typhus also OX19. Rocky Mountain spotted fever, this is OX19 as well as OX2. In case of scrub typhus, it is OXK strains they give the agglutination reaction. And in case of trans fever, none of the strains they give positive reaction. So this well-Felix reaction we can perform as in a tube, or can be used as a slide test. So this is very important test, diagnostic test for use most in most cases. It is used for diagnosis of rickettsial diseases. So coming to the treatment and control, as these organisms are intracellular, immunity has to be from both the arm of immune system. It is cellular and humoral, both arms. So vaccines are so far not available for rickettsial diseases in case of animals. Then some antibiotics we can prescribe, tetracyclines and chloramphenicol. They are shown to be effective in Q fever. Then the biological control of ticks or eradication of ticks is found to be shown to be helpful in the controlling of the rickettsial infections. Now, this is what I wanted to discuss today about the Rickettsia organisms. So I hope this information will be helpful, and if you find these informations are helpful, please subscribe to my channel, give your comment, and share this channel to with your friends so that everyone will be benefited from the informations I have compiled. And and please give your comments so that in our next subsequent discussions we can improve further to include more and more uh, uh, informations regarding the other organisms, as well as or other topics whatever we are going to discuss in our subsequent discussions. So with this, thank you very much, and bye bye.